The initial studies that were done with gene therapy for metachromatic leukodystrophy were done in Europe, and there's a significant amount of data that was accumulated over time there. So it's actually approved in the EU. So patients can get licensed therapy, gene therapy, for metachromatic leukodystrophy in the UK and throughout Europe. It's not been available as therapy in the US though. So we've been working with the company that is developing this and is responsible for this, which is Orchard Therapeutics, which is not my company. But we've been engaged with them about how we might be able to bring this to the patients in the US. And there's no open trial yet, but because they have a vector and they have the manufacturing capabilities, we wanted to offer for patients that we think might benefit the opportunity to get gene therapy in the US. So each one of those patients is treated under a specific IND that we'd have to put together for the FDA and get through all the regulatory groups and make sure that the company thinks it's a reasonable candidate for therapy. So it's a fairly involved process, and we have to tell the FDA that we would continue to follow these patients for an extended period of time, for 15 years actually. They want that long-term follow-up data. We've treated 3 thus far. There's the opportunity to treat more patients if we can identify them in a situation where they're likely to benefit. So that door is still open. The presentation was reasonably limited because we've only been doing this for about 1.5 years. The patients were all treated relatively recently, so we don't have a lot of long-term follow-up. But at least the data that we have thus far is quite encouraging. So there is very little toxicity associated with going through the process, certainly compared to allogeneic transplant, which is what we would otherwise offer them. The enzyme levels are at least normal, which is encouraging as well. And the follow-up that we've been doing shows that the vector copy number, the number of cells that were effectively transduced with the virus, seems to be very stable as well. So it's early, but we're very encouraged with what we've seen so far.